Okay, so hello everyone, welcome back to our Agreed platform, and this will be the lecture 2 for the genetics that we'll be going to deal with theories of evolution. So am I clearly audible to you all? And you can just see my video and the presentation. All the three things are visible to you all. Just give me a confirmation once.
Yes, sir.
Okay, my audio, video, and the presentation. All three things are fine, right?
Yes,
Okay.
sir.
Is there any background noise that is disturbing you? I have switched on
No,
the
sir.
I have switched on the fan. If it is creating disturbance, let me know. Okay. Okay, fine. So in the previous class, we have discussed about the history of genetics and cytogenetics. So where we have just divided the time periods into 4 different timelines, starting with before 1860, which is pre-Mendelian era, and after that we have. Okay, I'll just. So just to recall the things that we have covered from the previous class, it was dividing the total history into 4 different timelines. One is before 1860, that is pre-Mendelian era, and next it is 1860 to 1900, and 1900 to 1944, and 1944 to till present. So we have divided the total history into 4 different timelines, and we have discussed the major events that has occurred in these 4 timelines. I hope this is very much clear to you all. So in today's class, we'll just discuss about the evolutionary theories. Before that, I have few important terms that we can discuss now, and then we'll go to the theories. Okay, so starting with, I have two important terms for you. One is forward genetics and reverse genetics. Can anyone tell me like what is the difference and what does it forward and reverse genetics means? By the term itself, you already know that it is opposite to each other. The meaning is just opposite to each other. That's fine. But what does it mean? Like what is forward genetics and what is reverse genetics? Yes, anyone? So in the forward genetics, it is a study in the genetic itself where we have the phenotype. Yes, we have the phenotypic trait, or there's simply a trait or a phenotype. From the phenotype, we'll just go on searching for a DNA sequence, or we'll just isolate or extract the DNA sequence from the phenotype. So we have visibly looking traits. From that, we'll just try to find out the DNA sequence which is responsible for that trait. So we are going from phenotype back to the DNA sequence. So that will be the forward genetics. Yes, Ramya is answering it right. So the reverse genetic is just opposite to it, where we have already isolated the DNA sequence. So we have to find the function of this DNA sequence, or simply the stretch of gene. We have already a stretch of gene. We need to find what does it like, what is the function of this DNA sequence. So that is called as reverse genetics, where we move from DNA sequence to a trait. So these are the two important terms in case of genetics. Am I clear? If I'm clear, just raise your hands.
Yes, sir.
Okay, fine. Next, we'll discuss about the model organisms. Can you just list out the model organisms in case of genetic experiments that you study in the different classes, starting with bacteria, plants, animals? Just name some model organisms in which most of the experiments have been conducted in them.
Drosophila,
Yes, one is Drosophila. Okay. Drosophila,
Agrobacterium tuminifera,
Elena Gester, Agrobacterium. Okay. P. Okay. Mice.
Guinea pig.
Good. Guinea pigs.
E. coli,
Okay. E. coli. What else? Have you heard about this plant, Arabidopsis?
Yes, sir.
maize. Then we have bacteriophage. What else we have? Maize, Arabidopsis, bacteriophage, Drosophila, E. coli. Anything else? So these are the most common experimental material that are used by the several scientists, out of which the model organisms are actually Drosophila, E. coli, Arabidopsis, maize, and bacteriophage. So all other organisms like that we have listed are frequently used in the experiments, but the model organisms are the above I listed. Okay, so let us discuss like why they are the model organisms. We'll just start with the Drosophila. What is it? It is a fruit fly, right? So why it was the model organisms and what are the characteristics that made it very ideal for use to use it as an experiment material? What is it? Yes, we left another one which is neurospore. Yes, thank you, Anusha. So what made to use this as a experimental material? What are the ideal characteristics of this fruit fly? Yes, very good. It has short life cycle.
Large
Observable
chromosome.
characters.
Yes, more
Observable
number of
characters.
yes observable characters. More number of variations are there that can be easily identified, easily identifiable phenotype or the traits. Okay. What else? Contrasting, yes, the easily identified which are contrasting with each other, which we can differentiate clearly. So there can be clear, we can differentiate the traits into clear-cut classes. So that is what contrasting characters means. And mainly they are gone by like we have the traits like in every organisms that are gone by qualitative and quantitative. So we only concentrate on the traits which are qualitative. So that gives us the contrasting traits or contrasting characters. Okay. So these are the like what about generation time? You just left the most important point. So the generation time of these food flies is just 2 weeks, which makes it as a ideal experimental material. So along with it, so it can provide large number of offsprings in a single mating. So it will save a lot of time and energy and the resources that we use, right? So large number of offsprings in a single mating. And what about the maintenance? It is very easy to maintain. So the resources that is required is also very less and it is not even harmful. So these are the list of things or the characters that makes it very ideal for to use it as experimental material. So did I leave anything in this Drosophila? If you can remember any other characters which is very important. And regarding the chromosomes, also the fruit fly has a chromosomes which are very large chromosomes which make it very easy to observe in case of microscope. Okay, and lastly it's about the mutant strains. So there are several naturally occurring mutant strains in case of Drosophila, which was very much helpful to see the variation and study the variations and identify the gene sequences. Okay, anything else? Did I talk about the expensive? The okay, it is inexpensive. Even it is inexpensive. So all these traits, all these characters made it a very ideal organisms for to use it as an experimental material. So this was regarding the Drosophila. Secondly, we'll just talk about E. coli. The same similar characters like generation time. What is the generation time for E. coli? 2 weeks, 2 months, 2 years. What is it? Or 2 minutes? Yes, it is 20 minutes. So the generation time in case of E. coli is just 20 minutes. So this is the first and foremost important character or the characteristics which make it very ideal to use it as the model organisms for most of the experiment. So generation time and easy to like maintain or the easy to culture. The culturing is very easy. So this is the second point. Okay. And genetic manipulation. So what is this genetic manipulation? Addition, deletion, or modifying the genes is comparatively easy in case of E. coli since the genome size is very simple or genome itself is very simple. So it is very convenient to manipulate the genes and just observes the variations. And in case of E. coli, so there are many fundamental biological process actually simulate with the higher organisms. So whatever we study in the E. coli, whatever changes that we observe, so that can be directly compared with the higher organisms. So that is why it is one of the reason why it is the model organisms for most of the experiment. And lastly, since it is non-pathogenic, so it doesn't have any harm on any of the organisms. So this is the ideal one. Clear? Am I clear?
Yes, sir.
Okay. So we have discussed two, and third one we have Arabidopsis. Arabidopsis. So this is the model organisms in the plant kingdom. So why it is so? So again, it is because of short life cycle and small amount of genetic material. And then we have the similarity. So when we talk about the similarity, the genetic similarity of Arabidopsis will correlate with most of the crop plants or most of the other plant species, similar to which I have said in the E. coli. So that is the third point. So what else? Small size of genetic material and shorter life cycle. And again, it is because of clear and distinguishable traits. All these things are very much easy to understand, easy to remember. So you can expect these type of questions in either statement based or assertion reasoning. So make sure you don't miss the marks if they are asking any question regarding the model organisms from any of the type, whether they ask for E. coli, Arabidopsis, or the Drosophila or any other organisms. Okay. So next, we'll talk about maize. So maize is also one of the model organisms when we talk about the crops. So in the whole plant kingdom, the Rhodopsis is the model organisms, but when we speak about the crops, the edible crops we can take. So the maize is the one important or one of the model organisms. So why it is the model organism? Sorry for the disturbance. Hello? Yeah. So passcode I have sent in the WhatsApp group. You just check the WhatsApp group. I have sent the passcode and the meeting ID in the group itself. Okay, I'll just send you again. So I'll just send it in the general group. I'll just send it in the paid group also. Okay. 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 Sorry for the disturbance. So I was talking about the maize. So why it is the model organisms? First and foremost, regarding the genetic manipulation. So when I talk about the genetic manipulations, what about the male and female inflorescence? Can you just name the male and female inflorescence in case of maize?
Tassels.
Tassels.
Tassel
Oh.
and silk. So the genetic manipulation is very much related to these two different organs. So can you just tell me why it is so? So genetic manipulation, when we say it means that crossbreeding or crossing between two different plants. So when we just have to cross the plants, we have to consider the male and female inflorescence. But in case of maize, detasseling is very much easy. So if you are having the practical exposure, you might be already knowing that plucking out the tassel is very much easy, and there won't be any contamination if we do it very carefully. So that is the main reason why the maize can be the one of the model organisms for the most of the experiment. Clear? And that is what the experiment versatility means. Like I've just mentioned this in the notes, just go through the notes once. And second reason is about the rich diversity, since it is grown from the ancient times. Like there is a rich diversity and it has gone through the series of evolution. So that is why we see the rich diversity in case of maize plants. So that is very much helpful for all the experimental studies. And again, it is because of the grain. So have you seen the maize grains? We have the plenty of traits in the grain itself. So all these traits are very easy to identify and easy to distinguish. So how many of you are aware about mobile genetic elements, which I discussed yesterday? What is it? Mobile genetic elements. What is the other name? Mobile genetic elements, transposer
Transposons.
elements, insertion sequence. So Barbara, it was in the first time. See, so most of the discoveries have been made in the prokaryotes for the first time. And after that, they were just compared it with the eukaryotes, and then that is how we have come to know about many things. But in case of mobile elements, it was for the first time which was discovered in the eukaryotes. It was in the maize, the mobile genetic elements. It was first time discovered in case of maize, which was the eukaryote. So this was possible because of easily distinguishable traits in case of grains. Clear? So you're getting like what I'm trying to explain.
Yes, sir.
Okay, fine. So this is all regarding the important model organisms. So next we can just jump into the three important milestones which I was talking about in the yesterday's class. So there are three important milestones that you should be well aware of if you are studying the genetics. Milestones in case of genetics. Can you just tell me if one or two, if you know it, or you can just guess what are the major events which actually shaped the study of genetics?
DNA,
Okay. DNA, what is it? DNA double helix model, the structure, or we can take it as DNA structure, the discovery of the gene, or the preparing the DNA structure, or identifying the DNA structure and identifying the genetic material. Identifying the DNA as genetic material and decoding its structure. It is the one important milestones out of three. So one is already decoded. So what are the rest two? So
Black operon.
you left most important one in case of genetics.
Mendel,
Yes, see, without the Mendel experiments, would be not known about the heredity, or we would be not having any idea about how the characters will pass from generation to generation. So the Mendel experiments are very much important. So that is all where the birth of genetics took place, right? After its rediscovery. The Mendel laws are one important thing. Please mute yourself if you're unmuted. Okay. So first and second thing are done. So there are chromosomal theory of inheritance, everything they are the important one. But when we talk about the milestones, so these are the two. So what about the third one?
Black operon.
Lack of operon is one thing, but collectively it is comprehensive analysis of genetic material or the heredity material. Just someone said like lack operon we have, and we have done the linkage studies, the mapping, the advanced studies like DNA sequencing. All of this will come under the comprehensive analysis of genetic material. So these are the three important milestones in case of genetics, for in which you should be all like you all should be well aware of. So if someone asks you about, just tell me the major important or the three important milestones, or what are the important milestones in case of genetics, you can name these three, and then afterwards you can take a number of events that are very much important. Clear? Am I clear?
yes, sir.
Okay. So next we'll just discuss about some important terms. Okay. So we'll just start with inheritance. So how do you define this inheritance? So inheritance is the transmission of genes which governs the various characters. So that is what inheritance means, which passes from generation to generation. It is just a transmission of genes from generation to generation, which consists of like which are reasons for the various traits, or which are responsible for the various traits in the organisms. That is inheritance. So how do you define the character? Character might be the morpho, physiological, anatomical, or behavioral feature of any organisms or any organism. So this is how we can define the character or the trait. Third, we have genetics, which I have already discussed. It is study of heredity and variation. So what is the term, the Greek term for genetics?
Genetic code.
Geneticos, which means the genesis or generate or originating from, right? Next, we have cytogenetics. Now you should tell me what is cytogenetics and what is cytology. What is the difference between these two, the cytology and the cytogenetics? Yeah, give it a try. Anyone?
Various aspects of chromosome and their effects.
Various aspects of chromosomes and their effects. So this is cytogenetics.
Yes, sir.
Okay, good. What about cytology? Study of various aspects of chromosomes and their effect is cytogenetics. Very good. Cytology is the study of cell, while cytogenetics is study of chromosomes. Exactly. So here in the cytogenetics, it is mainly the study of chromosomes. Here it is study of cells. Okay. So to give a proper definition, the cytogenetics is a study of chromosomes and their effects on a development of various characters. So that is what cytogenetics mean. So proper definition for cytogenetics. So when we talk about cytology, cytology is a study of structural and functional organization of any organisms. It is a study of structural and functional organization of different structure you can take in a cell. Not organisms, you can take it as cell. Okay, the structural and organization, like functional organization of various structures, like a various different structure. In case of cell, it is cytology. Clear? So mainly you can, if you want to explain it in a single word, it is a cell which comes under the cytology, and it is the chromosomes which comes under cytogenetics. So what, sir, are the traits and the characters are same? So who can answer this? Traits and the characters.
Morphological, anatomical, and physiological behavioral of an organism.
Yes, but the question was, yes, you are right. It is a definition for the character. So what I am asking is, Ankit is asking whether the trait and the character are the same words or the synonyms, or they have the different meaning. Who can answer this?
Same.
Okay, same. One is answering it as same. So any other answers?
But both are different. Whether it's correct,
Actually, Vipul, your voice is breaking. We are not able to your voice is breaking. We are not able to properly hear you. You can just put it in a chat box. So, okay. If you are not answering it, just go today and just search, do your analysis and research, and just come back and tell me like whether they are same or different. And it's in confidence in answering this question. So this is a basic thing in genetics that we are studying. The character and the trait. Is there any difference or they are same? Okay. Just try to remind me in the different, just try to remind me in the next class. So no, they are different. Okay. Pranjal is answering it as they are different.
Excuse me, sir.
Yes.
Trait is alternative form of the character.
Yes, trait is the alternative form of a character. So what is character? Character we already explained, right? Just like
Trait
the
is what we see, sir.
Okay, character. What about the character? Okay, let me not make you more confused. Let us take one complete day and just go and just search again. So search from whatever sources you have. Just come up with a concrete answer. What is character and what is trait? Already few students are answering it. Let me just give a chance to everyone. Okay. Fine. So this was about the few important terms of genetics. Now let us just start with the evolution theories. So before the Mendel proposed his laws, there were the evolution theories. So most of it, the evolution theories that we have known are less based on the scientific evidence, less based on the scientific evidence, and they were popularized mostly because of the supporters and the oppositors. The opposition and it got popularized. The basic criteria of why only few of the theories got popularized, and mostly some of the theories got rejected by many of the biologists or biochemists. So it was mainly because of the supporters, how many supporters the theory had and how many were actually opposing the theory. So mostly and less, it was less basically with the scientific evidence and mostly with the popularity that the theories got. So that is how they came to know before the Mendel's laws were published. Okay, so we'll just discuss all these theories now. Let us start with the theory of spontaneous generation. So who proposed this theory? This theory of spontaneous generation, if you remember. Yes, spontaneous, like this is the most common theory that you might be hearing from your class 10th, the theory of spontaneous generation. So who proposed it? So what does this theory tells us? So it does simply means that all the living organisms have arose from the non-living things. So that is what the theory of spontaneous generation tells us. So it was
Louis Pasteur.
Louis Pasteur actually disproved this theory. He didn't
Radiant
support it.
hallucinate.
Huh? Yes, now this answer that as right, and it's Aristotle. Okay, if you Google it right now, if you just Google the same thing, that who proposed this theory of spontaneous generation, you might either get Pasteur or Reddy and Spelling. But these three are the scientists or biologists who actually rejected or who actually provided the experimental proof against this spontaneous generation theory. And it was initially by the Aristotle, and there were many supporters for this theory. And mainly the name is one of the most important name will be the Aristotle. Okay, so what does it theory says that all the living organisms are actually arose from the non-living things. So that is what this theory explains. But it was initially for the first time disproved, or the experimental evidence was provided by Reddy and Spelling. So what is the full name of Reddy and Spelling? If you know it, Spelling. Disproved. Theory of spontaneous generation was for the first time disproved by Reddy and Spelling. So what is the full name of these two scientists? Yes, so it is Francis Reddy and Lazzaro Spallanzini. Okay, I have already noted this in. Yes, very good. Ram has answered it right. Lazzaro Spallanzini and Francisco Reddy. So I have just given these names in the notes. You can just go through it. Okay. How many of you have downloaded the notes that I have uploaded yesterday? Just raise your hands. Okay, 17 people have done it. Okay, fine. So there are some few additions that I have done it today. So please, again, once I intimate you in the group, you can again download the new one. Okay. If you have already printed the notes, then you can just print the extra things, or you can just write the extra things that I have given in the new notes. Fine. So for the first time, it was disproved by Francisco Reddy and the Spallanzini, and then the final and the concrete evidence was provided by Louis Pasteur and the Tyndall. Sorry. So it was done independently. They both independently provided the experimental proof. The Tyndall experiment, so how many of you remember? It was using the cotton plugs. So they used the cotton plugs in the flask and just left it for several generations or several time. So just to see whether if there is any living organisms that has been arose from the non-living thing. But finally, like even after several generations, nothing came out of this flask. So that is how they provided the proof against this theory. Tanzila, kindly share the PDF of first and second lectures in the group. So I have already did it in the course. You can find the all the notes in the course itself. Okay. It was okay. Theory of spontaneous generation. I hope this is clear. So when we talk of theory of spontaneous generation, you have to remember three important scientists: Aristotle. Aristotle. You can leave the Aristotle, but you have to remember the three important scientists like Francisco Redi, Pesaro Spallanzani, and Lazzaro Spallanzani, and Louis Pasteur and Tyndall. So four different scientists. And what does this theory of spontaneous generation explains? So that's it for that. That is what is important for theory of spontaneous generation. Okay. So next we have second one. It is regarding the reproduction of plants and animals. So this is not a single theory. These are the series of events which actually contributed for the clear-cut understanding about the reproduction in case of plants and animals. I will just list out the events. For example, it was in 1673, de Graaf, he for the first time told that the tiny vesicles that we see in the mammals are actually the eggs. He just discovered the eggs as a tiny vesicles in the form of tiny vesicles in case of mammalian ovaries. And next, it was 1785, where the Spallanzani, along with disproving the theory of spontaneous generation, he also proved that the presence of sperm was actually essential. So it is regarding the reproduction. The sperm is very essential for the development of progeny. So this was proved by Lazzaro Spallanzani in case of 1785. And next we have 1875, where the conclusive evidence that fusion of sperm and the egg nuclei in sea urchin. So who gave this? So
Oh, Hertwig.
yes, it's Hartwig. Fine, very good. So next it's Grew. So what is the contribution of Grew? He just described the stamens in case of flowers. Okay, Grew. And next we have another name, which is Camararis. So what is the contribution of this Camararis? If you remember. So he showed that in a maize plant, he didn't get any he didn't get any seeds when there was no pollen. So he just gave or described the importance of pollen in case of formation of the seeds. So he showed that the seeds will form without the presence of the pollen. So that is his contribution. So what about the Fairchild's mule? So what is the year?
1717.
Yes. So this is one of the most common and important year to be remembered. So make sure to remember it. Okay, very good. Anusha has answered it, 1717, fine. So it is a cross between and the scientist?
Carnation
Thomas?
and William.
Sweet William.
Yes.
Sweet William.
Yes. Yes. Sweet William and Carnation. Okay. So that was called as Fairchild Mule. Next, in 1823, we have Amici. So what is the contribution of this Amici? Have you heard about the pollen tube entry into the ovary? So he described about the entry of the pollens, the pollen tube or the pollen tube entry into the ovary. So he described this thing, the Amici, in the case of 1823. And lastly, we have another important scientist who is again Strasburger. So what is his contribution that we have discussed in the previous class? It was regarding the
Fertilization
chromosomes.
in angiosperms.
Yes, here it is fertilization in angiosperms. So I just wanting you people to remember from previous class. What was his contribution that we discussed in the previous class?
Discover thread-like
Discovery of chromosomes.
structures during cell division.
Yeah, be more specific. Someone is answering. Yes, can you just repeat?
So discovery of chromosome.
Discovery, be more specific. I told you in the previous class, right? I just gave you the clear idea about all these things.
Discovery of all structure
We
in
refer chromosomes
ovule.
as thread-like structures.
Thread-like structure, sir.
Yes, very good. You have to remember this thing, because if you just remember the chromosomes, but you might be again confused with the four different scientists, because four different scientists revolve around the same term. So make sure to remember exactly what it is. So the thread-like structure, at least just remember the keywords. It's the thread-like structures that he described or discovered. Fine. So this is in 1884. So all of these events are very much related to which gave the better understanding about the reproduction process in case of plants and animals. Okay, so coming back to the theory, we have another important theory which got rejected. It is preformation. So who gave this theory?
Aristotle.
Okay. Any other names? Aristotle. Okay. Actually, it was originally proposed by Swaminathan and Bonnet. The full name I've given, it is James Swaminathan and Charles Bonnet. So there is short story here. I can make it short for you. So there are, again, in case of preformation, there are two different categories of people. So one category of people were called as animalculist. Animalculists. Okay. And second category were called as ovists. So if you just remember, can anyone tell me like what is the difference between these two category of people? So basically they see this the animalculists are the set of people who actually believed in this theory, and the ovists, they are the category of people who believed in this ovist theory. So this is the difference, and it was majorly led by Aristotle and Harvey. So animalculist means like they believed that majorly the offsprings are developed, it is because of the semen, and only the nutrition is provided by the females. So that is what they believe in. So that is why the name animalculist. In case of ovist, so they believe that like progeny is mainly because of the ovary, and the semen only provided the stimulatory effect. So that is what the ovist believed in. So these are the two different assumptions that were made, and they were the two category of people. Am I clear? The ovist and animalculist.
Yes, sir.
Okay, so this is under the preformation theory. Okay. Collectively, so all these supporters of this both the groups, what they believed is, so the miniature of the human or the miniature of the progeny is already present in the individual. So, but it is not complete. It is just a miniature, but it is present in the egg or sperms, and that will go into the progeny and that it will develop there. So that is what they believed in. But later it was easily disproved with the help of the microscope. So from the microscope in the egg and sperms, they observed that there was no miniature that was visible in either the sperm or the egg. So that is how this third theory was rejected. Clear? And it was called as homunculi. Sorry. Homunculi. So the miniature which I was talking about, they named it as homunculi, which was already present in either egg and sperm. It passed from the parent to in progeny and it developed. So this is what they believed in, the preformation. It is already preformed. So the progeny is already preformed before it is like it has given birth, and it just passes from generation to generation. That is what they believed, but it was easily disproved with the use of simple microscope. Simple microscope means not actually is a simple microscope with a simple device, which is the microscope. Am I clear?
Yes, sir.
Okay, so next we have epigenesis. So who gave this epigenesis theory?
Wolf.
Okay, again the full name. Wolf. Wolf is the right answer. What is the full name of? Yes. Come on. Caspar Friedrich Wolf. and it is F and F. It is not Wolf. It's Wolf. Okay. Caspar Friedrich Wolf. He gave the theory epigenesis. Can you just explain like what does this epigenesis theory tells us? Yes, anyone?
Tissue and organs are
Embryonic
originated
tissues.
differentiation process.
Okay. Sperms are eggs are undifferentiated. Okay. Any other answers?
Embryonic tissues consisting of cells which further differentiate into various organs.
Okay. Any more answers? If anyone is typing, I can wait. Organs are formed by differentiations in the embryonic cells. Okay. So initially the Wolf suggested that the organs and tissues will be developed through the process of just the differentiation. So it is just the already the small tissue is there by the set of differentiations. So it will be developing into a tissue or organ. So this is what Wolf initially explained. So later his famous successor was Van Boer or Van Baer. So he told that so this development of tissues and organs are because of sequential modification of the embryonic tissue. So both talked about the embryonic tissue, but he directly said that it is just because of differentiation. But later his successor Von Baer suggested that it is because of sequential modification of this embryonic tissue. That is how we see the developed organs and tissue in a completely developed man or woman. So this man or woman or any organisms. So this was explained or added by Van Baer. So this is most commonly or universally accepted fact now. This is just the theory of epigenesis. Next, we have one more important theory, which is inheritance of acquired characters. So who gave this theory?
Lamarck.
Lamarck.
Okay, not again, not again, not again. And it's the complete name. Yeah, it's good like you already prepared all these things, but just add these names to your preparation, okay? Jean Baptiste Lamarck, the full name. Okay, so he proposed the theory of acquired characters. So he told that before that, this particular theory was originally suggested by Hippocrates. It was in 400 BC itself. So the history reveals that this theory was actually proposed by the Hippocrates in 400 BC. Later, it was like popularized by the Lamarck. So what does this theory explains us? The characters of the individual will develop due to the changes in the environment. For example, if there is any organisms which has any like lost any trait or gained any trait because of the prevailing environmental conditions, so that particular trait will be passing from generation to generation. If for example, let us take it as mice, the mice, so if we cut the tail of the mice in this generation, the upcoming generation will also have tail less mice. Is it true? In the present generation, we just we just isolate one mice and cut its tail and we just reproduce it. So we get a tailless mouse. Is it true? Yeah. So this
No,
is about
sir.
this is about the theory of acquired characters. So this was again disproved. But most importantly, this theory was supported by Charles Darwin. Since he was working with the natural selection, he observed that due to the environmental conditions, the prevailing conditions, the traits or the characters will change, or there is an evolutionary process where it will be all these traits will be changing because of the conditions. So he was observing this natural selection. So that is why like he actually supported this theory or the idea of inheritance of acquired characters, but later it was disproved. Next we have organic evolutions. So what is this organic evolution theory? If anyone can tell me.
Survival of fitness.
Yes, organic evolution theory explains about the survival of the fittest. So it was proposed by Charles Darwin. According to him, so if there are different categories of within a single species, if there are different categories of animals or different categories of organisms, so out of the different categories, only the best one or the fittest one will survive and the weakest one will be extinct. So this is what the theory of organic evolution explains us according to Charles Darwin. So now tell me whether this is completely true or not.
No.
So can you just explain me how it is? Then what is true in case of organic evolution? Or we know that natural selection is a universal phenomena that will occur in nature. So how much is true in that and what is true in this theory, the organic evolution theory? So here he clearly told that if for example, there are two category of, let us take as animals. There are two category of animals. So if in that two category, if one is fit and one is weak, the weak one will be extinct and the fit one will be growing. It will be surviving. This is what according to Charles Darwin. But in the real case, what happens is both the category will survive. Both the categories will survive. But when we talk of number of progeny, the fittest one will have the more number of progeny and the weakest will have less number of progeny. So this is the difference, because this natural, why it is so, why it is so that two different categories will also survive, because the natural selection is not the qualitative one. It doesn't make the one group of characters extinct. It is quantitative, and the change that we see because of natural selection is gradual, not the sudden. There is no drastic change that you can see in the characters. It is gradual over the generation to generation. So that is what the quantitative means. So that is why when we just take the two different groups of categories of any organisms, both survives, but the fittest one will have more number of progeny, will have a more reproductive success when it is compared to the weakest one. Am I clear? So this is just a modification of it doesn't mean that this organic evolution theory is wrong, but it was being modified later. So based on our understanding about all these theories, we have just modified the things that will suit us. Fittest one will produce more progeny. Is it clear?
Yes, sir.
Okay. So any other theory that I have left with, if you remember? So we have started with what? Preformation we have covered, reproduction in plants and animals, and then we talked about organic evolution, theory of acquired characters. So what else?
Germplasm theory.
Yes, germplasm theory. Before that, we have one more important theory. Have you
Pangen.
heard of? Yes, it's about pangenesis theory. Again, it was proposed by Charles Darwin. So what does this theory explains us? The pangenesis theory. So it is mainly based on the organ, that every organ produces the identical copy of itself. So that later it will be transferred to the progeny and it will be developed into the organisms. So that is what this pangenesis theory explains us. Or the organ, the identical copy were called as pangene or gemmules. Is there any similarity between this pangenesis theory and theory of acquired characters? Yes. So if you just compare the theory of acquired characters and pangenesis, there are similarities where whatever changes that we observed in the present generation, the same changes will be passing on to the next generation. This is what both the theories explains us. But one theory tells us it is because of the environment. The pangenesis doesn't take the environment into consideration. It just explains that the organ will make its identical copy. That will be the gemule or pangene, which will be transferred to the next generation, and it will develop into new individual. So this is about the pangenesis theory. So who rejected or who disproved this pangenesis theory that was
Weismann.
proposed by?
Weismann.
Okay. Before that, there is a cousin of Charles Darwin. So who was the cousin of this Charles Darwin? Have you heard this name? Francis Galton. Have you heard of this name, Francis Galton? No. Okay. So he was the first person to challenge his own cousin theory, which was put forward by the Charles Darwin. So after that, it was the August. What is the full name of Weismann again?
August.
Yes, it is August Weismann. So he disproved the pangenesis theory by putting forward the germplasm theory. So since the pangenesis theory talked about the organs that make its identical copies, so this Weismann clearly explained with the new concept of germplasm, where all the tissues are divided into two different types. One is somatoplasm, and second one is germplasm, where the characters or the changes that occur in this germplasm will only pass to the generation because it has the egg or the sperms. But the changes that will occur in case of somatoplasm, that won't be transferring into the next generation. So out of these two different categories or two distinct types of tissues, only the changes that occur in the germplasm will be transferred from the individual or the parent to progeny, not the characters of the somatoplasm. So what is the difference between somatoplasm and the germplasm? If anyone can answer me.
Somatoplasm do not have
Plasm
any
means it
influence
is
on
the
the
total genes that we have been
reproduction.
collected.
Okay, one by one.
So somatoplasm do not have those tissues do not have any influence on reproduction, sexual reproduction.
Okay, so these do not contribute for the sexual reproduction. So when we talk like so basic difference, if the most common difference, if you ask me like what is the difference between somatoplasm and germplasm, it is in case of reproduction. So this is a basic difference. So except this reproduction, there is no other any other fundamental difference that you see. So the difference is only in terms of the production of gametes, where the gametes are specifically produced by the germplasm tissues. Somatoplasm do not produce any gametes. It is only the mitosis that occurs in the somatoplasm. Clear? Yes. Rituraj has answered it. Somatoplasm is mitosis, germplasm is meiosis. So in case of germplasm, is it only the meiosis or the mitosis can also occur in the germplasm? So it can occur. Okay, Anushka is answering it can occur. Rituraj just told me that somatoplasm is mitosis. It has a mitotic behavior, and meiosis behavior is shown by germplasm. Yes. So if you don't remember it, just make sure to remember it's both mitosis and meiosis. See, without mitosis, none of the tissue can develop or none of the tissues can divide. If from the meiosis, if the number of gametes or the genome size is reducing from generation to generation, then it will go extinct, right? So it is always the meiosis followed by the mitosis when the gametes are formed, okay? Or the mitosis event will occur and then the meiosis event will occur. So this is how it will carry the information from generation to generation. So if I want to put it in words, somatoplasm, it is mitosis, germplasm, it is mitosis plus meiosis. So this is the fundamental difference between these two distinct types of tissues. So can you just name any organisms which doesn't have the distinct somatoplasm or germplasm?
Unicellular organisms.
Yes, unicellular or single cell organisms. They do not have distinct somatoplasm and germplasm, and the microbes, you can say, which are mostly the unicellular. So these do not have the distinct somatoplasm and germplasm. Okay, so previously I told you that the somatoplasm are not inherited or they are not involved in reproduction, right? So while I was discussing about the fundamental difference, I told you that somatoplasm does not involve in the reproduction. But who proved this? Who proved the fact that somatoplasm does not involve in the reproduction? It was by
Phillips and
Castle.
Castle.
Yes, very good. It was by Castle and Phillips. So there is a one simple experiment that I have already given in the notes. You can just go through the experiment. It is very easy to understand, by which you'll come to know it is just a evidence for what he has put forward. The theory that he has put forward, that okay, somatoplasm doesn't involve in the reproduction. So simple evidence he has provided, you can just read the evidence. Anyhow, that is not important, but it's very simple. You can understand it. Anything else that I have left? So finally, it is the germplasm theory which is universally accepted from all the different theories. Pieces, okay. So you all are clear with whatever I have discussed till now? Any doubts?
Clear, sir.
Okay, if there are no doubts, I have a few questions for you, which I told you in the yesterday's class, that I will be just showing you a few questions from the previous year papers that you can answer now. So these are the questions from the previous year papers. Mostly questions from the first chapter came in 2020 paper and 2021 paper. So answer this question. So let us start with cell theory. Cell theory is related to which scientist?
Schleiden-Schwan.
Schwan and
So it is A is for 2, okay. B, chromosomal theory of inheritance.
Saturn
Dunn
and Bovary.
Walter Sutton and okay. Divisibility of gene
Oliver.
that we have not discussed. Yes, it's Oliver.
Oliver.
One
Ingram.
gene, it is Ingram. So in yesterday's class, after the one gene, one enzyme hypothesis, I was talking about the I told you, like, if there are any other hypotheses that came forward, it is one, that is one gene, one polypeptide. Okay. After one gene, one enzyme hypothesis. So it was put forward by Ingram. Just remember this. So D is 3. Okay, next. Just read the two statements and give an answer. I told you about the 3 components of cell theory. So can you just please recall it for me? What are the 3 different theories? Just write it down.
Omnicellular, acellular.
Okay, omnicellular, acellular was put forward by the scientist.
Virchow.
Yes, it was.
Rudolf Virchow.
It was Rudolf Virchow. Then, so
Every
what
new
are
cells originate from pre-existing cells.
okay? It was about the pre-existing cells. So what is the next one? So the structural and
Structure
functional
and functional unit of
Cell.
organism. This was Schleiden and Swan, and it was by Rudolf Virchow. So that is what they are asking. So you people are getting the idea, like how the question comes in the exams. So we have discussed all these 3 points. You have to apply it while you answer these statements. So this is the 3 components where these what they are talking about, which I mentioned here. So these 2 statements are correct. And again, the match the following. Mostly you can see the questions from the first chapter, which are match the following type. Sorry, I got cold today. Central dogma of the molecular biology.
Francis
That I will
Crick.
that I will be discussing in molecular genetics. No need to worry. So you just match the other things. Again, it is chromosomal theory of inheritance. Yes, it's Francis Crick. Okay. Second one, chromosomal theory of inheritance.
Walter Sutton and Theodore Bloom.
See, the questions are actually repeating. You can see the things here. So these are the most frequently asked questions. Double helix model, it's James Watson and Crick. And fourth one gene, one enzyme hypothesis, it is Biddle and Todd. Okay, see the full names. They are giving the full names. Yes, George Walter Sutton, like George W. Biddle and Edward Todd. Okay. Clear? Next, we have again the mask following. So in this one, the discovery of human blood group we have not discussed, but I hope like you'll be knowing this answer. It's Landsteiner. Anyhow, we'll be discussing in upcoming classes. So what about P, DNA, DLX model? Again, it is C. They have added the Willikens here. Crick, Watson, and the Willikens, which we have discussed in the last class. Why the Willikens was included, why he was contributed, how he contributed for the DNA double helix model, right? So it is B for 2. And next, the genetic improvement in Mexican wheat. It was by Green Revolution. It is all about the Green Revolution. It was Norman E. Borlaug. Next, mobile genetic elements in maize, which I have discussed. It's McClintock. So D for A. Okay. So these were the questions that you can see from the chapter 1. So this is the importance. See, if the questions have been asked from the chapter 1, you should not miss the question in by any chance. Okay, this is the most easiest questions that you can that can appear in your ICAR JRF exam or any other state entrance or CUET exam. Okay, make sure not to commit any mistakes if you get these questions in the exam. Is it clear? So how many of you attended the mock tests that I have uploaded yesterday? Just raise your hands. No one has attended the mock test. Okay. So just attend the mock test today. Within an hour, I'll be uploading the updated notes. You can download it and you can read it once. Then you can attend the mock test. Fine.
Sir, it was not opening yesterday. I just entered.
Okay. So there is some problem in case of app. So it is updating. I have just complained it to the developer. We'll be doing it in 24 hours, I guess. Today, again, I complained him regarding the same. There was a time error, but everything will work fine in the website. So if you want to attend the mock test, just go through the website. Just search for agility.in in case of any browser. From there, you can attend the mock test. Okay. It is just for 1 or 2 days. Again, the app will come live and you can conveniently use our mobile application for all these things. Is it fine?
Yes, sir.
Okay, fine. So we'll meet in the next class. We'll be just starting with the chapter 2, which is cell ultrastructure. So in the tomorrow, even it is Sunday, since we have started from Friday, we'll take the class on Sunday. So we'll be starting with chapter 2, which is cell ultrastructure. Fine. So we'll be finishing or winding it off in a single class, and then we can move forward for the Mendelism. So if there are no doubts, we'll meet in the next class. Very good night to you all. We'll meet you soon. And if you have any doubts, you can directly post it in the group, WhatsApp group. If you are not registered our course yet, kindly register our course. Just take our student referral program very seriously. And I didn't tell you about the offer, right? So if you are referring your friend, he will get 5% extra discount on our course. If you just take your name, like the students who have registered, if you just refer someone, he will be also getting 5% additional discount with the 5% discount that we have already provided. Totally, you will be getting 10% discount on 3,500 rupees, the actual course price. And you will be getting the 200 referral amount. Okay. So just please refer this course to all your friends. If you're finding this course very helpful for your ICAR JRF plant sciences preparation or any other exams like CUET state entrance, if it is helping you, just suggest our course to your friends. Any doubts?
No, sir.
No doubts. Okay. Yeah, doubts won't come now. Doubts will come in the later topics, which are very interesting. Okay, we'll deal it. Okay, fine. So if you face any problem accessing the course content while through the app or the mobile app or the website, just feel free to contact me directly. I'll be helping you with all the things. So if there are no doubts, we'll end the session and we'll meet in the next class. Good night, everyone. Bye-bye.
Thank you, sir.
Okay, thank you. Thank you so much for attending. We'll meet in the next class.